Hi everyone. Have you ever wondered how to choose the right rotation speed for a meaningful dissolution method? Why do we use a specific RPM and how do we justify it scientifically? Rotation speed is not just a number in a dissolution method. It's critical scientific decision that can make or break method performance. In this sixth video of Dissolution Science series, we will decode how to select agitation speed confidently and scientifically. In dissolution testing, hydrodynamics means the shear stress and flow pattern around the dosage form. Rotation speed directly controls boundary layer thickness and therefore dissolution rate. Agitation defines the hydrodynamic environment the dosage form experience. If the speed is too low, you risk under dissolution, and if speed is too high, you may mask real formulation differences. That's why choosing the right agitation speed is critical. It controls hydrodynamics, influences dissolution rate, determines discrimination between formulations, and impacts method robustness and reproducibility. Right RPM means meaningful dissolution data. Rotation speed directly links to dissolution kinetics. Think of the boundary layer as a thin wall hugging the tablet surface. When rotation speed increases, this wall becomes thinner, allowing the drug to diffuse faster into the medium. In simple terms, drug dissolution depends on diffusion layer thickness. Higher RPM, thinner boundary layer, faster dissolution. The relationship is explained by Noyes-Whitney equation and Nust-Brumer model. That's why RPM selection directly controls dissolution rate and must always be scientifically justified, not chosen by habit. Rotation speed is not universal. It depends on formulation behavior. For multi-particulate system, lower RPMs are often preferred because excessive agitation can cause floating, sticking, or erratic movement. The right rotation speed must match how the dosage form interacts with the hydrodynamic environment to generate meaningful and reliable dissolution data. RPM selection depends on dosage form type. For example, IR, MR, DR, ODT, or multi-particulate system. API solubility. Formulation characteristic. For example, hardness, coating, or density. Apparatus hydrodynamics. For example, basket type or pedal type. Regulators generally prefer using the lowest rotation speed that still provide acceptable discrimination. These RPM values are not fixed rule, they are starting point guided by product behavior, industry experience and regulatory expectation. Typical range includes apparatus 1 basket 50 to 100 RPM, apparatus 2 pedal 50 to 75 RPM, ODT and multi-particulates 25 to 50 RPM. Poorly soluble drug often need higher RPM for meaningful release. Highly soluble drugs use moderate RPM to avoid over dissolution or coning. The goal is always the same. Choose an RPM that is scientifically justified, discriminatory, and clinically meaningful. Lowest effective RPM is the best RPM. pH adjustment can improve drug ionization and solubility, but it must remain physiologically relevant. Extreme pH values may boost solubility, but they can reduce clinical relevance, so a careful balance is essential. A practical RPM strategy is start with moderate RPM, typically 50 or 75. Observe formulation behavior, sticking, floating, or coning. Evaluate 2 to 3 RPM to identify discriminatory power. Select the RPM that maintains sync conditions and avoids hydrodynamic artifacts. In short, choose conditions that are scientific, realistic, and reproducible. Balance solubility, hydrodynamics, and relevance for meaningful dissolution data. I'll share a real world example without breaching confidentiality to show that many dissolution problems are actually hydrodynamic. In practice, adjusting RPM is often the simplest and most powerful fix. Common examples: coning, increase RPM, for example 50 to 75; floating, switch to wasket or reduce RPM; sticking to the vessel, slightly increase RPM or adjust the medium. Excessive variability, try lower agitation to stabilize hydrodynamics. Over-sensitive methods reduce RPM to create a less aggressive environment. Fix the flow before fixing the formulation. Dissolution is a quality control tool, not just a release test. Regulators expect discriminatory methods that can detect meaningful formulation or process changes, such as tablet hardness, coating variations, or particle size differences. In many cases, lower RPM provides greater sensitivity, making it easier to see real differences between formulations. Using an RPM that is too high can mask formulation differences, which creates regulatory risk. That's why RPM selection must be scientifically justified during method development. Right RPM protects product quality and regulatory confidence. Always retain photographs and raw data to support regulatory justification. This example shows how a structured RPM comparison leads to scientifically justified selection. 50 RPM, floating observed not acceptable; 100 RPM, dissolution too aggressive, no discrimination; 75 RPM, no artifacts, stable hydrodynamics, and good discrimination. That's how RPM should be selected, by evidence, not by habit. Data and visuals strengthens your regulatory defense. When properly optimized, it makes your dissolution method robust, reproducible, and regulatory ready. A practical approach is: begin with mid-range RPM, avoid hydrodynamic artifacts, choose an RPM that clearly differentiates formulation changes, provide clear scientific justification in the final method. Remember, regulators prefer scientifically defendable choices. Thank you for watching. Please subscribe us for getting update on such industry application oriented videos, and stay tuned because in this around 62 video series we will dive into dissolution method development, validation, troubleshooting, advanced approaches, and real industry case studies. In the next video we will cover dissolution profiles and F2 similarity factor, a quick guide. If you have any question, please post them in comment section. I will do my best to respond. See you in next video. Thanks.